Hello, my name is Angelina Allen, and my Module 4 local expert project is on cryogenic electron microscopy. To begin, let's talk about what cryo-EM is. In short, it is a technique used for protein analysis and visualization, as we have discussed in class. This method is used to make detailed high-resolution atomic 3D images of molecules and complexes using cryogenic freezing and electron microscopes. Cryo-EM can provide detailed images and information on RNA, viruses, proteins, cells, mechanisms, functions, and more. Cryo-EM has been used in microbiology, immunology, neurobiology, and many more. It has been used to look at viral infections and bacteria, to visualize protein aggregations and neurodegenerative disorders, to study multicellular organisms, and more. Next, let's look at the basic workflow for cryo-EM. The first thing that needs to be done is to put the sample in a grid. The purified sample is placed on a grid usually made of metal like copper or gold and blotted with filter paper. The grid is then either cryogenically frozen by plunge freezing, where it is dunked in liquid ethane that was cooled in liquid nitrogen, which is called cryogen, or it is frozen by high pressure freezing. Smaller samples typically use plunge freezing, whereas larger samples use high pressure freezing. This freezing puts the samples in a glass-like state that prevents the formation of crystals and protects the sample from being damaged. Next, the sample is placed into an electron microscope where electron beams are passed through the sample to get 2D projections on the detector. These 2D images are then layered and averaged in computer software to create a 3D map and model of the sample. There are two types of cryo-EM, and they are both used for different things. Cryogenic transmission electron microscopy, or cryo-TEM, is used to look at the internal structure of the sample, and it uses a transmission electron microscope to do this. The sample is hit with a beam, and the interaction with the electrons results in scattering and absorption. There is a projection of the image onto a detector, and the sample is tilted to capture multiple angles so a 3D image can be generated. This method is used for single particle analysis and can show molecular structures and interactions. Cryogenic scanning electron microscopy, also called cryo-SEM, is used to look at surface and topography of a sample and uses a scanning electron microscope. The sample must be fractured or cleaved to expose the inside of the sample, and the microscope creates a focused electron beam that scans the surface of the sample. Electrons are emitted as this interaction occurs and detected to create the 3D image. This method is used for larger structures and for surface characterization. Next, let's talk about some of the advantages of cryo-EM. First, the cryogenic freezing of the sample can reduce the radiation damage as much as 6-fold compared to traditional electron microscopy. The advancements on this technique have also made it faster and more efficient so that it can compete or even replace other methods like X-ray crystallography or NMR. This method also has near atomic high resolution that allows the small details of samples to be obtained. Additionally, this method can be used on a variety of different molecules, including proteins, viruses, cells, and more. This technique can also be used on larger samples compared to some other techniques. Finally, the freezing itself helps preserve the sample so it can be studied and keeps it in its native state. Although there are many advantages to cryo-EM, there are also some limitations. First, smaller samples may be difficult to use because it might be hard to find them under the electron microscope. The fracturing and cleaving required for the samples for cryo-SEM can also leave physical defects on the sample, making it difficult to image. Additionally, the freezing process is very laborious and samples can be damaged during it. The samples also require homogeneity for there to be high-resolution 3D models made. This process compiles large data sets, and these can be computationally demanding. Finally, this process is expensive and requires highly skilled personnel. A lot of the controls for cryo-EM depend on the type of sample being used, but here are two that are common for most cryo-EM experiments. Negative staining is a heavy metal salt stain that is used for visualization because it has a high contrast to allow for the sample to be assessed. Negative staining is used to assess sample purity, concentration, heterogeneity, and conformational flexibility. The choice of buffer for this technique is also very important so that it doesn't damage or affect the sample. Buffers with phosphate, detergents, reducing agents, or glycerol can cause damage to the cells. Additionally, some buffers can affect the quality of negative staining and reduce specimen contrast. This figure is from the paper Cryo-EM Structures Reveal Coordinated Domain Motions that Govern DNA Cleavage by Cas9. This paper was using cryo-EM to see different states of the Cas9 endonuclease, which gave new insights into the domain motions and interactions between the enzyme and nucleic acids. This figure is the cryo-EM structures of 3 states of the Cas9 complex. The images in C through E are density maps of Cas9, and the images F through H are the schematic representations of state 1, 2, and 3 of the complex. The protein colors in the 3D images correlate to the protein domain shown in figure B. This figure is from the paper Assembly of a Nucleus-like Structure During Viral Replication in Bacteria. In this paper, they used cryo-EM to show that phages assembled a nucleus-like structure in the bacteria during infection that separated viral DNA from the cytoplasm. This figure shows the cryo-electron tomography of the phage compartment during infection of Pseudomonas chlorophyus. B shows a segmentation of the sample showing extracted structures such as the shell in purple, the capsids in green, and the outer membrane in red. C through G show the progression of an empty capsid to an assembled phage, and in E through G you can see the viral DNA being assembled in the nucleus-like structure. This figure is from the paper Three-Dimensional Structural Characterization of HIV-1 Tethered to Human Cells. This paper used cryo-EM to look at the tethering-mediated restriction of HIV-1 because it is not well understood. Tethering is a host mechanism that prevents the release of enveloped viruses by linking the virions to the plasma membrane. This figure shows the spatial arrangement of multiple tethers on mature HIV-1 virions. The gray pictures are the tomographic slices, and the colored pictures are the manually segmented tomographic reconstructions of the mature HIV-1 virions. The tethers are in red, and the plasma membrane is in cyan. Here are the sources I used if you want to learn more about cryo-EM. Thank you for watching my presentation, and let me know if you have any questions.